So what is antibiotic resistance and why are we so concerned with it? Antibiotic resistance can mean a variety of different things in terms of genetics to bacteria, but for all of these different mechanisms, it just means that these bacteria are going to survive if they're exposed to an antibiotic. So in the case of penicillin, as we have been talking about, just means that penicillin might be around, but it's not going to end up causing the break in the bacterial cell wall that we had discussed previously. Antibiotic resistance means that bacteria will no longer be susceptible to an antibiotic. So again, remember that susceptibility means that they are inhibited by an antibiotic. If bacteria have antibiotic resistance, then they are no longer susceptible to an antibiotic. So as I said, this can mean a variety of different things for bacteria in terms of what they're actually doing. And they can, you don't need to know all these, but they can be actually modifying the chemical structure of the antibiotic, as we see here. So penicillin can be inactivated when bacteria have certain versions of genes for antibiotic resistance. In other cases, we have a reduced permeability, which it sounds like it affects the cell membrane, but it actually affects these proteins in the cell membrane. So the antibiotic would be normally bound by some receptor on the cell surface. Again, that receptor is a protein. It's not necessarily there to bind to the antibiotic, because why would a bacteria produce receptors that are going to make it, susceptible or vulnerable to a threat? But this would be some protein that the bacteria uses to sense and interact with other signals in the environment. So if this bacteria were to become antibiotic resistant, you could see the shape of the receptor has changed slightly, and it will no longer bind to the antibiotic. So something to keep in mind for this type of antibiotic resistance is that this is a protein. This is some type of molecule that the bacteria is using normally, and it needs it to be functional. And so when it changes the shape, you have to question, does this make it less functional for its normal use, for what the bacteria is using it to sense in the environment besides antibiotics, for example? And the question or the answer might be that this reduces the effectiveness of this protein for other functions. And so that would be a reason why bacteria would not want to get this antibiotic resistance, especially if there are no antibiotics in the environment. So other varieties of antibiotic resistance would be that the bacteria actually gets a new gene that is for a protein that pumps out the antibiotic. So it does get into the cell, but it's not going to remain in there for very long, so it doesn't really hurt the cell. And then there are other versions of what we talked about on the previous slide for changing different molecules. So effectively eliminating the binding site for antibiotics. And I don't mean getting rid of the binding site entirely, but just changing it again so that the antibiotic is not able to bind. So we see here, would be the antibiotic. Again, here we're talking about changing the ribosome. The ribosome is very important, right? We can't produce any proteins if our ribosome is not functional. And if we lower the efficiency of our ribosome, then that means we're making proteins more slowly. So that's not great. And in other words, again, bacteria are not going to want this gene for antibiotic resistance, this gene for modifying the ribosome, if they don't need to use it, if they're not exposed to antibiotics. And again, this gets into things like evolutionary theory and natural selection, which we're not going to talk too much about, but just keep in mind that there is a trade-off here. But it's worth it if there are antibiotics in the environment. And then finally, there are similarly other ways that bacteria might avoid having binding sites, so using alternative pathways for proteins, or again, just changing the shape of the protein. So why are we so concerned about antibiotic resistance? Well, I mean, that's pretty obvious, right, guys? Antibiotic resistance means that you cannot easily treat bacterial infections. And if we have even a few cells which are antibiotic resistant, then they can pass on that DNA to their offspring. And here we see how selection can occur. So we just have a few cells that are antibiotic resistant here. And then if the person takes antibiotics, but there are these few cells, then what could happen is that all the other cells are going to be eliminated. They're either going to die off or just not pass on. Their genes are not going to divide. And then that leaves only the progeny, only the offspring of the bacteria that have genes for antibiotic resistance. So now the whole population is resistant to antibiotics instead of just having a few cells. If, as we kind of talked about previously, if the person is not taking antibiotics, if maybe they have a very mild infection and they say, well, you know what, I'm going to see if I could just get over this, then these cells that don't have antibiotics, that don't have antibiotic resistance, are going to take up all of the resources, potentially. And as we said, they might be more efficient. They might be better at dividing. Their ribosomes might work better, or their cell wall enzymes, cell wall building enzymes might work better and faster. And so they're going to outcompete the ones that have antibiotic resistance. And so this would lead to just hopefully a healthy person. This guy, it's not really looking like a person, but that's okay. And then he is healthy. He's gotten over an infection, and there's no antibiotic resistant bacteria. And again, that works if you have a mild infection that your immune system can deal with itself. Another reason why you might not take antibiotics is if you have a viral infection. So that's why doctors are being a lot more careful nowadays about differentiating between viral and bacterial infections, so that you don't get this phenomenon from a viral infection where you're not going to even end up killing off the infection from taking the medication. All right, and then one other thing that we should talk about is the fact that, as we've talked about previously, bacteria can share their genes, right? And so these guys that have antibiotic resistance, as we see here, highlighted in red again, they could pass those antibiotic resistance genes on to other cells, and then those can pass the genes on to other cells, and so on. So then potentially you could end up getting a lot more individuals in the population that have antibiotic resistance, even if you don't kill off all the other bacteria. And this can occur through horizontal gene transfer. And we sometimes abbreviate that HGT, and that can occur through a couple of different mechanisms that we talked about previously. Conjugation with a pilus, transduction, and it could also occur through viral transfer. A lot of these antibiotic resistant genes can occur on plasmids, and so we usually depict them like this. So a plasmid, again, is a small circular piece of DNA, relatively small. It's smaller than a genome, but some plasmids can still be huge. They can have thousands and thousands of base pairs. And so what happened in the between 2010 and 2015, I think, we got things like Mrsa, and these bacteria had plasmids that carried multiple antibiotic resistance genes. So they carried genes that made them resistant to multiple antibiotics, not necessarily all through the same method, but through different methods. And this plasmid can just be shared because of the HGT that bacteria are so good at. And so I mentioned Mrsa, and so that is MRSA, and it stands for Methicillin. Sorry, this is messy. Methicillin resistant Staphylococcus Aureus. And so it's just a variety of staph. Again, it's not its own species or anything. It's just this species and certain cells of this species that happen to have a plasmid that gives them resistance to methicillin and potentially other things.